So this is lecture 15 review. There are 3 questions that are going to be theoretical, meaning there's no Punnett squares involved, no having to problem solve and work things out. You are going to have 60 minutes for this quiz because most of the questions you are going to have to work out using Mendels laws, using the Punnett square. But of the 3, you're going to have to essentially know the difference between Mendels law of segregation and Mendels law of independent assortment. And then you have to know the relationship between genes and traits. All of the other ones are going to be examples that we've talked about during the lecture, genotype, phenotype examples, as well as the examples of autosomal dominant, autosomal recessive, a lot of Punnett squares. So let's begin with the review on Mendel's laws and the genes and traits, and then we'll talk about all of the different Punnett square types of questions, and I'm going to visualize them over here on this canvas area for you. So Mendel's law of segregation basically is that doesn't matter what your 2 alleles are. You can be homozygous dominant, homozygous recessive, or you can be heterozygous, but it applies to everybody, and that is that all people have 2 alleles and they only pass on 1. So a child is going to be the combination of 1 allele from each parent. Now this will be important when we're looking at some of the other Punnett square questions on blood type and how different parents can pass on certain things. And those will be the application. But as far as the theoretical one goes, make sure you can define the law of segregation properly, as well as define the law of independent assortment, because that's essentially what I'm going to ask you is those definition questions. Independent assortment is the more difficult one. So let's look at this again real quick. We know that homologous chromosomes pair up and line up during meiosis 1. And we also know that genes can have different versions, different alleles of those, dominant, recessive. And so when you have different genes on separate chromosomes, the law of independent assortment basically says that you cannot predict the inheritance of one gene's allele when you know the inheritance of the other gene's allele. Basically, they don't influence each other. And so in Mendel's experiments, when he saw the round peas and the yellow peas, or the wrinkled peas and the green peas, he found that whether they were round or wrinkled and whether they were yellow or green did not influence each other. And that was because during metaphase 1, during this period of time where independent assortment occurs, because homologous chromosomes can flip to either side, in these scenarios it's showing you the different combinations that you can have, mainly that you can have the round yellow, or you can have the green wrinkled, or you can have the round green or the wrinkled yellow. And basically the shape of the pea does not influence the color of the pea. And that's because these genes are on separate chromosomes, and due to independent assortment, they assort themselves independently of each other. And that's, in human terms, if you have a parent who's brown-haired and is really intelligent and the other one is blonde-haired and not very smart, just because your child inherits brown hair does not mean they're going to inherit the intelligence genes, even though it's an oversimplification, obviously. But that's just kind of the idea, is that you can't say, oh, he's got my eye color, so he's probably going to be smart like me, or he's going to have my blood type, or he's going to have my strong immune system. That's not how it works at all. And that's what the law of independent assortment essentially says, is you cannot predict the inheritance of any other genes based upon the inheritance of one genes, because they're all spread out along these chromosomes and they all independently assort themselves on one side of the on the metaphasic plate. Okay, that's one question. So make sure you know the difference between the law of segregation or the law of independent assortment. The other questions are going to be relationship questions between alleles and traits. So from this list, dominant, recessive, incomplete, codominance, and from this list, polygenic, pleiotropy, genetic heterogeneity, and epistasis, these are the concepts that are going to show up as the answers. I'm going to define one of these concepts, and you tell me which one I'm talking about. So again, just review back over what each one of these mean. What does it mean to have a dominant allele? What does incomplete dominance mean? The 2 that people usually mix up on the exam are incomplete dominance and codominance. Incomplete, remember, is when you get a mixture of the phenotypes. Curly plus straight equals wavy. Red plus white equals pink. That's incomplete dominance. Codominance was what we were talking about with the blood type. This is the only example of codominance I'll ever give you. And that is people who are blood type AB are basically both phenotypes. They're blood type A and they're blood type B. So blood type AB is actually two phenotypes. And that's what codominance is. Again, coming back to this scenario, if, and I stress if, the red and the white were codominant, you wouldn't have pink petals. You would have some red and some white. You would have both phenotypes at the same time, right? I don't even know what it would look like with the hair. You'd have some hair strands that are straight, some that are curly. You'd have a bad hair day every day. Anyway, so those are the theoretical ones. Let's go back and jump into the Punnett square ones, because I know that in the video I talked about it, but you really weren't able to see those examples. So let's go through those examples now. So the first example that everyone's going to have is a generic genotype-phenotype question where I generally use just big A, little a. I'm just essentially going to use the big A as a dominant allele, little a as a recessive. We're not doing any disorders or traits or anything of that sort. And so I'm going to set it up on the exam saying, this parent is homozygous dominant, this parent is heterozygous. What are the chances that they'll have a child that is heterozygous, right? So you'll set it up by saying, okay, one parent is homozygous dominant, the other parent is heterozygous, right? We're doing the Punnett square here. These are the two alleles that the parents could pass on. And so when you put the Punnett square together, the combination of those alleles essentially gives you this distribution. So if in that question, if I said one parent was homozygous dominant and one parent was heterozygous, this would be the outcome. And you would say, oh, it's a 50% chance that the child would be heterozygous. Now, there's another version. There's actually many versions of these questions, and this is also going to test you on your ability to understand what phenotype is, because I won't just ask you what the genotypic ratio of this is, but I'll ask you possibly the phenotype. So pay close attention to the phrasing of the questions, because I may give you the same question and say, what is the chances that the children will have the same phenotype as the parent? Well, in this scenario, it's 100%. The reason for that is because this parent has the dominant phenotype. This parent has the dominant phenotype because they have one dominant allele, one recessive. And all the children have the dominant phenotype, whether they're homozygous dominant or heterozygous. So in this scenario, it would be 100% dominant, meaning the phenotype of the children would be the same as the parents. Now, in other scenarios, I may say both parents are heterozygous. What are the chances that they'll have a child who's homozygous recessive? Or what is the distribution of their alleles? There's many different ways in which I can ask you the same question. So if we set up the Punnett square and you put together the alleles from each parent, right? Remember, the little a in this column, little a in this column. So when you put these alleles down these columns, you get this combination. One question may say, what is the allelic distribution? What's the genotypic distribution? I usually say genotypic distribution, and this would be 25% homozygous dominant, 25% homozygous recessive, and 50% heterozygous, okay? If I ask you what the phenotypic distribution is, you would say 75% dominant, because these three right here, homozygous dominant and heterozygous, those are all dominant phenotypes, and this is 25% homozygous recessive, or the recessive phenotype. So 75% dominant, 25% recessive, that would be the phenotypic ratio. Genotypic ratio, 25% homozygous dominant, 50% heterozygous, 25% homozygous recessive, okay? So I have about 3 different scenarios. Usually I'll have two parents that are heterozygous, one that's homozygous dominant and one's heterozygous, or one that's homozygous recessive, one that's heterozygous. Those are kind of the 3 different scenarios I give. And this is just to test you on your understanding of terms like genotype, phenotype, heterozygous, homozygous. So everyone will get one question like that. So that's one of the questions you're going to get. Let's look at some of the other ones. So there are there's one question on an autosomal dominant disorder as an example, and one question on autosomal recessive disorders as an example. Now, typically I use Huntington's disease or I use achondroplasia as examples on the exam, generally because I use concepts like Marfan syndrome on other hypothetical scenarios, or polydactyly and whatnot. And so I tend to favor Huntington's disease and achondroplasia. On autosomal recessive, I tend to favor cystic fibrosis and sickle cell anemia. Again, sickle cell anemia is an autosomal recessive, even though it's not showing up on this list. Doesn't matter which one I give you because they're all essentially going to be the same. So let's begin with autosomal dominant inheritance. So coming back to our Punnett square, let's go through some scenarios. The hardest part of doing any of these is really just setting it up. Once you set it up, it's really easy to figure out what the results are and what I'm asking for. So for Huntington's disease, remember that when we're dealing with alleles and genotypes, if you're homozygous dominant or if you're heterozygous, both of these lead to the disorder of whatever dominant disorder we're dealing with. So as far as Huntington's disease or achondroplasia, it doesn't matter which one we're dealing with here. Now, if you are homozygous recessive, this is what we call a normal phenotype, right? So if you don't have the disorder, then we would say that you have the normal phenotype. So that's terminology I use on the exam. So, for example, if I say one parent is heterozygous and has Huntington's disease and the other parent doesn't have Huntington's disease, let's set that up before I ask you what the results are. So the parent who is heterozygous, obviously, again, they've got one of each of the alleles, a dominant allele and recessive allele. The parent who doesn't have Huntington's disease, they have the normal phenotype. I didn't tell you what their genotype is, but I don't have to because there's only one genotype associated with that, right? So in that case, this parent would be homozygous recessive. Now, if you put together the combined effect of these two parents, then no matter what question I ask you, you've got it down. I can say, what's the probability that the children won't have Huntington's disease? Well, that's 50%. What's the probability their children will have Huntington's disease? Well, that's also 50%. All right, let's erase this real quick. Let's look at another scenario. So same thing, but this time both parents have Huntington's disease, and they're both heterozygous. Notice I have to tell you whether they're homozygous or heterozygous because there are two different genotypes associated with Huntington's disease or achondroplasia or whatever dominant disorder I give you, because just having one allele is enough to have the dominant disorder. So in this case, both parents have Huntington's disease and they're heterozygous. So this is how we would set it up. You combine these two and those two, these two and those two. So in this case, we would say, what is the probability that the children will not have Huntington's disease? Well, in this case, it will be 1 in 4 or 25%. What's the probability the children will have Huntington's disease? That's 75%. Notice it doesn't matter whether they have one dominant allele or two dominant alleles, they're going to have the dominant disorder. And so in this case, again, this is one of those things asking you about the phenotypic ratio. In this case, the phenotype is Huntington's disease, in which case it would be 75%. Okay. There is one that usually trips people up, so let's go over it. It basically does the reverse and says you have statistically from two parents, half of the children have Huntington's disease and half of the children don't have Huntington's disease. And most people don't know how to approach this one. So let's go over it. In this case, half the children don't have Huntington's disease. Well, we know what that genotype is, right? So let's just put that in right here. All right, let's work backwards now. Well, in order for the little h to go down this one, it has to be here. And in order for the little h to be over here, it has to come from this parent, right? So we know that part of it. Well, statistically, the other half of the children have Huntington's disease. Well, that's kind of easy to do then at this point. That parent has it, and when we combine the effect of those two, now you know what the parents are. And so if I give you that question where I say half the children don't have Huntington's disease, the other half do, what is the genotype of the parents? Boom, there you go. So you do have to work backwards sometimes. Again, it's just the same concept of the law of segregation. We're just giving you the statistics of the children, and you've got to work backwards to be able to tell us what the genotype of the parents are. Okay, so that shows up every once in a while. So I give you that one to make sure you know what's going on. Now let's switch gears to recessive disorders. This is the one that changes things up a little bit. Let's go back down to the bottom here. Again, if we're dealing with recessive disorders, it doesn't matter in any of these cases what letters you use. I like to use A because my little a's are way different than my big A's. But in recessive disorders, remember that you must have 2 recessive alleles to be able to have the disorder. So in the case of cystic fibrosis, sickle cell anemia, Tay-Sachs disease, fetal ketonuria, all of that, the child or the person must be homozygous recessive. Any of these other genotypes have the normal phenotype. Now remember that this is also a carrier. So if you are heterozygous, and I use the word carrier a lot, carrier only refers to recessive disorders. It does not refer to dominant disorders, because if you are a carrier, it means you have a normal phenotype, but you also carry the recessive allele. So that's essentially what heterozygous means for recessive disorders. All right, so if we do a scenario like cystic fibrosis, and let's say both parents are carriers of cystic fibrosis, how would we set that up? Well, carrier means heterozygous. So that's pretty easy. You basically combine these two, combine these two, combine these two, and combine these last two. So if 2 parents are carriers of a genetic disorder that's recessive, then they have a 1 in 4 chance of having a child who will get the disorder, right? All the other children will have the normal phenotype. So it's a 75% chance that they won't get cystic fibrosis, 25% chance that they will. Now this applies across the board, whether it's sickle cell anemia, cystic fibrosis, Tay-Sachs, it doesn't matter. That's why I just kind of do this generic A. You can use whatever letter you want when doing these Punnett squares. Okay, let's do another one. Let's say that one parent has cystic fibrosis and the other parent is not a carrier and does not have cystic fibrosis. Now notice I did not give you the actual genotype of that parent, but let's start with the first one. That parent has cystic fibrosis. Okay, we know what their genotype is because they have the disorder. But the other parent, I said they don't have cystic fibrosis, but that could be homozygous dominant or that could be heterozygous. Well, in the case of where I said they're not a carrier, boom, we just eliminated the heterozygous. So in this situation, that would be this parent right here. Now, what's the probability that these 2 parents will have a child with cystic fibrosis? 0. Okay, all of the children will be carriers, but 0 probability that they'll have cystic fibrosis. Now, there's another question, and this is the one that also trips people up whenever I give it, and so I'm going to go over it with you. The question is, one parent has cystic fibrosis. What is the probability that their children will carry at least 1 recessive allele? Okay, listen to that closely again. One parent has cystic fibrosis. What are the chances that their children will carry at least 1 recessive allele? Okay, well, you're like, well, I can't do it because I don't know what the other parent is. Listen to what I just said. It doesn't matter what the other parent is. The question is asking, what are the chances that the children will carry at least 1 recessive allele? The answer, 100%. Why? The law of segregation. This parent is guaranteed to pass on one of their recessive alleles. They're homozygous recessive for the genetic disorder. They will pass that allele on. I'm not asking you what this other allele is. This parent could be, a carrier. This parent could be homozygous dominant. That's fine. But I'm asking, what are the chances that the children will carry at least 1 recessive allele? So look out for that one as well. This is one that it gets boring if I just say, here's this parent, what's the probability? That really doesn't test your knowledge on genetic inheritance. So that's why I throw some of these questions in there to make sure that you understand exactly what's going on here. Okay? All right. Let's erase this. And now, sex-linked. This is the one that throws people off as well. And the reason for that is because the Y chromosome always trips people up. And that's why you got to be particularly careful. And when you do the Punnett squares, you might want to make them a little bit larger. And the reason for that is because draw the sex chromosomes. And the reason is because if you draw the sex chromosomes, you will not get mixed up with this. That's because women have 2 alleles for every gene on their X chromosome. Why? Because they have a homologous pair. Men only have 1 allele for their X chromosome because the Y chromosome has a completely different set of genes on it. So men are much more susceptible to sex-linked inheritance, especially recessive disorders, than women are, because women need 2 recessive alleles for both of their genes to have the disorder, be it color blindness or be it hemophilia. Those are the only 2 scenarios I give you on the exam. So again, draw the sex chromosomes, because if I say that a woman is a carrier of color blindness, so that she has 1 dominant, 1 recessive allele, and the man is not colorblind, a lot of times people will say, well, is he homozygous dominant or is he heterozygous? And they'll say, I don't know. Again, men only have 1 allele. So if he's not colorblind, then he has the dominant allele. If he is colorblind, it's going to mess it up a little bit here, but that's fine. If he is colorblind, he's going to have the recessive allele. Okay? So draw the sex chromosomes whenever you're doing any of these scenarios, because people always think that men have 2 alleles for the sex-linked inheritance. That way you're like, okay, there's no allele here on the Y chromosome, okay? If you don't do that, then you're going to get mixed up with some of the probabilities that I give you on the exam, okay? So let's do some example questions on the exam. So if I say that a colorblind man has children with a woman who is a carrier for color blindness, let's set that up here, okay? So the woman's a carrier for color blindness. She's going to have 1 dominant, 1 recessive. The man is colorblind, so that's his only allele. He's got the recessive allele, okay? So without showing the, all the others, if I go through the X here, we've got a dominant and a recessive, dominant from the mother, recessive from the father. And we've got 2 recessive from each of those. Down here, the males will just get the X chromosome from the mother, and they get the Y from the father. And in this case, the X chromosome from the mother and Y from the father. So this one's not too bad, because if I were to ask you what's the probability that the children will be colorblind, then it would be 50-50, no matter what the sex is. And that's not normally the case, right? Here, 50% of the daughters will be colorblind, 50% of the sons will be colorblind. Over here, these daughters will not be, and these sons will not be. So it's 50-50. But let's go through another scenario here. Let's reverse it. Let's say the mother's colorblind and the father is not. Again, this works for hemophilia as well. So the mother's colorblind. The only way she can be colorblind is if she's homozygous recessive. She has 2 alleles for her genes because she's got the homologous pair. Men, 1 chromosome, 1 gene, 1 allele. He's not colorblind, so he must have the dominant allele. So when we put these together, we find that none of the daughters will be colorblind. The reason for that is because they have basically the dominant recessive. But let's look at the sons. 100% of the sons will be colorblind. So this is where the statistics change, and this is why this messes everybody up on the on the quiz. So make sure you understand, again, how this works. Men only have one version of that gene. That Y chromosome has a completely different set of genes. It is not homologous. It doesn't have any genes for that. So whatever allele men get, that is the phenotype they're going to get. If they get the recessive allele, they have the recessive phenotype. If they get the dominant allele, they're going to have the dominant phenotype. So this one is a scenario where none of the daughters will be colorblind, but all of the sons will be colorblind. Okay, so keep a close eye on that one. That's one that trips people up quite a bit. Now, let's deal with incomplete dominance. This one's a pretty simple one. Incomplete dominance, again, comes down to the fact that sometimes there are no recessive alleles. Sometimes you just have dominant alleles. In this case, there's two dominant alleles, the red and the white allele. And when you mix them together, you get pink. And so this one's pretty simple. You just have to know which alleles are what. If I say, oh, a red flower is mixed with a pink flower, what's the probability of having pink flowers? Well, let's do that. We do big C for the dominant allele, but remember there's two versions of this. There's the R, which is the dominant, and the W, which is also the dominant, the red or the white. So one flower is red, and the only way it can be red is if it has the two dominant alleles. One flower is pink. Remember, this is the incomplete dominance. This is the intermediate phenotype. One red, one white. You get a mixture of the two alleles. So when we put these two together, we find that half of them will be red and half of them will be pink, right? So 50% red, 50% pink. Now, the example being shown up here in the upper right corner, that's what happens when you mix two pink flowers together. You get 25% red, 50% pink, 25% white. Okay, but I can switch this up and say, oh, we've got a white flower with a pink flower, in which case you'd get, 50% white and 50% pink as well. So this one's not too difficult. Most people get this one just fine, but just be aware again that this is that incomplete dominance one that we're mixing two different dominant alleles with. Now, this last one, this is where the remainder of your Punnett square questions come into play. We've already done a Punnett square on a generic one. We've done one on autosomal dominant. We've done one on autosomal recessive. We've done one on sex-linked, so those are 4. We've done one on incomplete dominance, that's the 5th one. You got 2 more, and that's all on blood type. Okay, so there's 2 different types of questions that I'm going to give you on blood type here. So let's go over the first one. The first one is a variety of different scenarios where I give you both parents' blood type and you got to tell me what the children's blood type is. So kind of like what we've been doing for all the rest. The difficult part of this is you have to remember that blood type O and blood type AB only have one genotype associated with them. Blood type A and blood type B, these are the phenotypes, they'll have 2 genotypes associated with them. So you have to make sure that when I say, oh, this parent's blood type A and this parent's blood type B, that I tell you whether they're homozygous or heterozygous. And I generally do all the time. I'm not testing you on this point whether you can figure it out. I'm going to give you the information, but you got to make sure you can put it together right. So, for example, if I say one parent's blood type A and it's heterozygous, they're going to have the dominant allele and the recessive allele. That's the heterozygous genotype for blood type A. If I say one parent's blood type B and heterozygous, they're going to have the B dominant allele and then the recessive O allele. And when you combine those together, that's essentially what you're seeing down there on the bottom right. You can get any blood type from this. You get equal proportions, 25% blood type AB, 25% blood type B, 25% blood type A, and 25% blood type O. Okay, let's do some other scenarios. Let's say one parent is blood type A, the other parent's blood type O. Well, blood type A, which one is it? Homozygous dominant, heterozygous. Let's say in this case, it's homozygous. Okay. So this parent is blood type A, homozygous. This parent's blood type O. I didn't have to say homo or heterozygous because there's only one genotype associated with blood type O. Okay. When you combine these two together, you essentially get 100% probability that the children will be blood type A, right? There's no other different combination. So if I say, what's the chances that their children will be blood type A? You'll say 100%. Okay, let's do one more scenario, and then we'll move on to the other question. So everyone will get some type of Punnett square question where I'm giving you the parents' genotypes, their phenotypes, and things like that. And then I'll show you what the last one is. So here, let's say one parent is blood type AB and one parent is blood type O. Well, if I say it's blood type AB and blood type O, there's only one genotype associated with each one of those. And so when we combine these together, you can see that you get 50% blood type A and 50% blood type B. Okay, so that's one of the questions. The other question is a little more difficult, and it's like what we were talking about before, where you're going to have to reverse the Punnett squares, because I'm going to give you the children, and you need to tell me what the parent essentially is. So let me explain. Here, let's say one parent is blood type A, and the other parent's blood type O, and the child is blood type O. I'm going to be testing you on your understanding of the law of segregation without really doing Punnett squares. So what am I talking about? Well, we know what the genotype of this child is. That's the only genotype associated with blood type O. Same thing with this parent. We know what the genotype is because it has to be blood little I. But this parent, we're not quite sure what they are. But we do, actually, because we know what the child is. So the question will actually say, what is the genotype of this parent? Well, we know that this parent had to have passed on one of their alleles. Didn't matter which one they passed on. They passed one of those on. But we also know that one of the alleles had to come from this parent. Well, that means that the other one has to be that because they're blood type A. So if I ask you what's the genotype of this parent, you know what it is because you know what the child is. Now, there's other scenarios that aren't as easy as this one. Let's say one parent's blood type A, one parent's O, and the child is A. Okay, well, let's figure out what we know. We know that parent. We don't know this parent yet. Maybe we can figure it out, maybe we can't. But we do know the child. Why do we know the child? Well, there's only two genotypes associated with this. Two dominant alleles or dominant recessive. We know that they had to have gotten one of the recessives from that parent and their blood type A, so we know that. Do we know this parent? No, we know this one. We don't know what that other one is. So on the question, if I say which of the following must be true, do not choose one that says that, oh, this parent is homozygous dominant or heterozygous. Choose the one that says the child is, sorry, I messed up on that. The child is A allele, O allele, basically heterozygous. That's the one you're going to be looking for, okay? So let's do that again with just some other scenarios here. Let's be funny here. Let's say both parents have BO, haha. And so let's say the child has blood type B. Okay, same thing. It doesn't even matter. It's the same scenarios I gave you last time. We know this. We know this parent. We know the child. We don't know this parent. We know that they have at least one allele, but we don't know what the other one is. So the child is going to be heterozygous. Okay, so if I ask you on the exam, which of the following do you know for sure? That's the only thing you know for sure. You don't know this parent. Okay, let's go through some other ones. Let's say that one parent is blood type A, the other parent's blood type B, and this child is blood type O. Okay, this one's actually pretty easy if you think about it. We know the child, and you know that each parent at least has that dominant allele. What's the other allele? Well, it has to be recessive. Why? The law of segregation. If the child is blood type O, he had to have received it one allele from each parent, which means that each one has to carry a recessive allele, in which case you would say, oh, both parents are heterozygous in their genotypes. Okay, so that's the last scenario I'm going to give you, where I'm essentially going to say, here's this parent's phenotype, here's this parent's phenotype, here's the child. What do you know? What can you determine from that? Okay, so do some of your own scenarios when you're doing this, but that's essentially what that last one is going to be. So Let's recap. You won't have anything on blood type like matching or other things like that. That's more just for general knowledge and everything. You're going to have 10 questions, 1 on Mendels Laws and 2 on the relationship between genes and traits, where the answers will be genetic heterogeneity, epistasis, pleiotropy, polygenic, incomplete dominance, codominance. Those will be the answers. I'll describe them. You tell me which one I'm describing. The other 7 questions are going to be the Punnett square questions. So it's essentially going to be the generic one on genotype, phenotype. It'll be the autosomal dominant inheritance, autosomal recessive inheritance, sex-linked inheritance, incomplete dominance, and then the 2 on blood type, the one that's just kind of general blood type from the parents, and then the other one where I give you their phenotypes and you have to tell me what their genotypes are, as I just went over. Okay, so that should review it for lecture 15, 60 minutes, twice as much time as you normally have to be able to work out a lot of those issues. Work out a lot of them beforehand. Then you already have them ready to go when I give you some of those scenarios. Again, give yourself all the advantage you can when going through this.